everybody. My name is Christiana Skarlatopoulou. I'm a scientist at CN Bioinnovations. And today I'm going to present to you NASH drug discovery using human microphysiological systems. Firstly, I'm going to talk about the overall drug discovery landscape regarding NASH. Then we're going to talk about the current preclinical tools that we're using for NASH. Then I'm going to give you some overview information on microphysiological systems, or MPS. We're going to be talking specifically on CN Bio's NASH MPS model, and obviously the drug efficacy in this NASH MPS model. And ending this presentation, I will have a quick summary of the main points that you should remember. Now, let's have a few definitions out of the way. What is NAFLD? What is NASH? NAFLD stands for non-alcoholic fatty liver disease, and it's characterized by the accumulation of fat within hepatocytes. Unfortunately, it affects 25% of the population, which is quite a high percentage if you ask me. Now, on the other hand, NASH is the progression stage of NAFLD, and it involves further aspects such as fibrosis, inflammation, and tissue scarring. 30% of NAFLD patients develop NASH, so again, a quite high percentage for that. And here to your right, you can see an overview of the progression of this disease from healthy to NAFLD and then to NASH, and the final stage, which is cirrhosis and end-stage liver failure. The interesting points for this specific schematic, I'd say, are the reversibility between the stages NASH and NAFLD, as well as NAFLD and healthy liver. Also important to know is that in current nomenclature landscape, we have some changes, and these include the names. For example, NAFLD is now being called MUFLD due to the addition of the metabolic dysfunction associated with fatty liver disease, and a similar thing is happening also for the NASH being converted into MASH, again, bringing in the metabolic aspect to this disease. Now let's have a look at the overall therapeutic landscape. Here to your right, you can see the various drugs and areas that the drugs belong to that are currently in the preclinical stage. And it's important to note that there is no drug on the market for NASH, or it has never ever been on the market. So there's been a lot of failure all these years, and there are some therapeutics currently that are in the late stage clinical trials. And what we can see here also from the diagram to your right, we see that the NASH pipeline is pharmacologically diverse, which also reflects the complex metabolic syndrome that manifests in the liver. Now, let's have a look at the NASH barriers for this, the barriers developing these therapeutic developments. So here to your right, you can see a schematic of how this process, of all the steps during the process, including the huge amounts of funds that go into this and the time frames that can be 15 years, for example, and some of the major phases such as preclinical phases, and target validation. In my opinion, the major barriers out of the 5 here that I've listed are the lack of preclinical tools that we currently have that fully recapitulates the disease spectrum that we see in humans, as well as the lack of mechanistic information to drug developers. Let's talk about the current preclinical tools that are being used so far. And these include the 2D cell culture, the in vitro 3D spheroid cultures, as well as the very classic in vivo animal models. As you can imagine, for example, the in vivo animal models, while they are complex, they do not recapitulate the human aspect of this disease. And obviously, the animal models are not humans. Therefore, the human relevancy is difficult to achieve. Now, on the other hand, the 2D cell cultures and 3D spheroids lack functionality of the liver and therefore the complexity that comes with the NASH phenotype. So as you see, there is a gap in the preclinical tools. Other considerations in wanting to develop a different and a better model for NASH in vitro are the new workflows are coming into play, such as new chemical modalities that are, for example, more relevant to humans. If you consider, for example, siRNA treatments, they're only targeted to specific human genes or other new chemical modalities. It's important also to consider that the access to NHPs are quite difficult and quite time-consuming, as well as not very cost-effective, that further bottlenecks this process into getting the therapeutics into the market. Another interesting change is the ethical concerns that have been raised globally about animal use in general. And you can see this also in the changes to the regulatory landscape, where a lot of countries and areas have pushed towards, reducing animal use overall. And one of the most famous ones is the FDA Modernization Act that happened in December 2022, which removed the mandate for animal testing at the preclinical stage and named, for example, MPS or organoid platforms as an example for other techniques. Some other countries included also South Korea, the European Union, India, and Canada. So let's recap what are the requirements to develop such preclinical models for in vitro use. Firstly, and most importantly, it needs to be human relevant. It needs to inform the design of animal studies. It needs to provide mechanistic insights into NASH. It needs to express a wide range of human translational biomarkers in order to be able to make informed decisions on the therapeutic efficacy. So how do we develop such a model? Let's get into the alternative method called microphysiological system. I've put out a definition for everybody to have a look at, but I'm going to stick to the equation that I've put at the bottom, which describes it, I think, more succinct, and that basically a microphysiological system is composed of two main things. And the first thing is some sort of cell type, tissue, organoid, and the origin can be human or animal, depending on what you want to model, of course. And then the other major factor is the microenvironment, which can include biochemical, electricals, mechanical cues, and that will generate this tissue organ model in the microphysiological system sphere. Now, microphysiological systems come in various shapes and sizes. And here to your right, you can see various schematics of systems that are currently in the market. Again, as I said, human cells and tissues are one of the main components, where you can see multiple of these models, schematics here on your right. Then obviously you have some sort of fluidic flow that will provide, that will mimic blood, will provide oxygen and nutrient supply, as well as some sort of biomechanical stimulus. For example, here in this one, you can see the chambers that the microfluidic flow will pass through for the cells to be able to survive. And then you also have some sort of environmental control, which can be mechanical actuation. For example, and here you see ECM gels are giving some sort of stability, as well as are helping with the communication amongst cells. And lastly, it's important that we have some sort of drug dosing or even sensors so we can control the therapeutic exposure. Because as we said, we want to use the microphysiological systems as a preclinical tool. That means We want to effectively model a specific organ or even a organ system, but also test drug efficacy on this model. Now let's talk liver on a chip. What are the crucial hepatic features that we need to recapitulate in vitro? So we have an accurate image of the liver niche that is happening in vivo. So it's quite interesting to note that the liver has a very interesting architecture in terms of how the cells organize themselves, which is in a hexagonal shape. I don't have a picture on that one here, but the three main cell types that you will have to consider specifically for NASH would be the hepatocytes, which is the main cell type in the liver, then the resident macrophages, which are called Kupffer cells, and finally the Kupffer cells, the hepatic stellate cells, which are important for the ECM modulations / generation. Now, in terms of important markers to look at for the liver, so albumin and urea are two main health markers to look at, and they're basically not only health markers, but also functional markers. So when these are expressed, that means the liver is healthy and functioning as it should be. And obviously, since we want to check for therapeutics, we always want to include some sort of marker for hepatic damage, because as we probably all know, the liver is one of the main detoxification organs in our body, and that transforms xenobiotic compounds from our body and can be the main organ to die in case of a toxic event from a compound. So these markers can include ALT, AST, ALT and AST, and LDH release, for example. And then another important feature of the liver, as I said, because of the detoxification properties that it has, we need to have a phase 1 and phase 2 enzyme expression. I mean, the main one that everybody talks about in regards to the liver is a CYP3A4, but there is an array of other ones, as you can see here on the list, that are quite crucial. Lastly, transporter expression is also quite important, and you can see a few of them here at the bottom, such as ABCC2. Now, let's have a look at the liver MPS that CN Bio has developed. Here at the top, you can see the schematic of our liver plate, which consists of 12 wells. In each well, you have here, as you can see, the liver 3D scaffold, which has multiple tiny pores where the cells form tissues. Here is a schematic, a cutout out of the schematic of a single well, where you see where the liver scaffold sits, and you have the microfluidic flow, which is driven by the micropumps here, where you basically have the circulation, so to speak. Here is a further close-up of the 3D scaffold, where you see the individual pores and where the cells sit inside of these pores and have formed the tissues. And obviously, you also see the microfluidic flow going down up and generating also the O2 gradient. Now here to your very right at the bottom as well, you see the whole system. So this plate that I've shown you before sits within a driver that docks onto a docking station within the incubator. So basically this whole thing here goes inside of the incubator, and then we have the controller here, the big chunky machine, goes outside, and you can control fluidic flows and set up your experiments from there. Now, here you can see how the tissues form over a course of 7 days. Here at the top, there is a section of a whole scaffold. It's only a section, it's not the whole scaffold here you can see. And at the bottom one, we can see a single pore. And you can clearly see that there is a nice morphogenesis from single cells into a nice microtissue within the individual pores. Now, how do we know these are functional, though? As I said, we need to check for specific markers of the liver. And here, if you check the confocal imaging that we've done, we've stained for albumin, which I've previously mentioned already is quite important health and functional marker. And you see that it's clearly expressed. Another characteristic, even though we do not have cholangiocytes included in our system, is the architectural formation of bile caniculi. And finally, as you might know, the hepatocytes are epithelial cells, which means there is a need of tight junction formation, which is again here shown quite clearly. What we've also done is compared our system, our liver MPS, with other preclinical tools such as the spheroids and sandwich cultures. And as you can see in both of them, in the metabolic activity as well as the albumin production, for both of them, the liver MPS performs significantly better than the other two culture types and remains quite stable over the course period of 20 to 30 days, depending on the graph that we're currently looking at. Now, let's dive deeper into the NASH MPS model. So as I said, again, we have the plate schematic here, and this is a the single well schematic at the bottom where we see the fluidic flow of the single well. And we obviously need primary cells, as I previously mentioned. So one thing that I haven't talked about yet is that not all primary cells will form the microtissues that I've previously spoken about. So what we do at CN Bio is we validate the primary cells that we receive from vendors. And this is an exhaustive process where we firstly start with the validation of the monoculture of primary hepatocytes. Then we co-culture it with Kupffer cells, and then we co-culture them with hepatic stellate cells. So it's a lengthy process that ensures that the microtissues form nicely, and we can have accurate and stable results across all of our experiments. Now, obviously, we need to induce this triple culture into the NASH phenotype, if not even genotype. And this we accomplish by adding dietary fat medium throughout an experiment, for example, of 14 days. And here's a nice confocal image you can see after 14 days of culture with the fat medium. And obviously, we need to be able to check for readouts that are relevant for characterizing the NASH phenotype, which includes steatosis, so basically lipid formation, lipid droplet formation, inflammation, fibrosis, and then obviously liver functionality needs to be checked throughout the experiment to ensure that the liver is still functional throughout the experiment. Now, here are some examples of a proof of concept, if you will, that we definitely see the markers of various cells being present, showing up. So here we have at the top the stellate markers. So we have low MPC and high MPC. So if we don't have a low number of stellate cells, we don't see the markers for stellate cells being as high, which is obviously makes sense. And a similar image is also happening when we add a higher number of Kupffer cells, which are the macrophages in the system. And then obviously here on your right, we see the fat loading across time by adding the fat medium. And if you look at the image here, you clearly see the fat droplets accumulating within our pores in the microtissues. Now, obviously, we need to check all these different readouts to make sure we're actually having the phenotype that we want. And here we have various markers to check that. And as you see, we've compared hepatocyte monoculture only and the NASH culture for these different markers. So here at the top, we have the inflammation panel, and here at the bottom, we've got the fibrosis panel. And as you can see, for both of these situations, when we have the triple culture and the added fat media, we have higher readouts for inflammation. So we have increased inflammation, which we already know from the clinic. So therefore, we definitely see that there is a comparison between those two. And the same happens also for fibrotic markers such as TIMP1 and procollagen that we see here at the bottom. Again, for the NASH phenotype in our system, we get a high readout for fibrosis, that as we would expect from the clinical data. Now, what we have tested here as well is to add a further fibrogenic factor, which is called TGF-beta here at the top. So as I already mentioned to you, NASH is already driving the fibrosis towards a worsening effect, basically more fibrotic markers to be expressed. But TGF-beta is a further, accelerator into that. And you can definitely see that when you look at the confocal imaging here, where we have increased signal for alpha SMA as well as collagen, two of the main fibrotic markers here. And even looking at the soluble markers of fibrosis, you see an increase in all of the fibrosis markers by adding the TGF-beta on top of the fat media. Now, I've talked a lot about phenotype so far, but it's quite important to talk about the transcriptomic and basically the genotype. And here you can see that the transcriptional profile of the PhysioMimix NASH assay is more closely matched to the profile of human liver tissue than the murine models. You can assess this by looking at these two scores here, where you see that the MPS is and the human NASH Venn diagram here, the crossing, so to speak, has a higher score than the murine NASH one over here. So that's quite important to note as a factor. Now let's finally look at the pharmacological response. So we have tried various drugs. Here we see the dose-responsive effects of elafibranor in the PhysioMimix NASH assay, which is a PPR alpha delta agonist, which was in phase 3 clinical trials, and then it was dropped because it didn't meet the efficacy standards set by the company, by the clinical trial, basically. And as you can, and important to note that these doses that you see, so we have 3 dosages here, and obviously the control that was not treated at all. The dosages match the clinical dosages. And as you can see, for all the 3 important features that we see, which is inflammation, fibrosis, and steatosis, we see that the highest concentration of elafibranor had a significant lowering effect of all these 3, which is very important because it matches the clinical trial outcomes as well. Now let's have a look at the different one, which includes obeticholic acid. Here you see again the 2 different concentrations of obeticholic acid, which are again the similar ones as they use in the clinical trial compared with elafibranor. And you see that both had an effect on the highest concentration specifically had an effect on collagen as well as alpha SMA, so mainly fibrosis markers here. And this is also verified here by the quantification of these images, where the highest concentrations of both obeticholic acid as well as elafibranor managed to reduce the fibrosis of NASH, of the microtissues that we built. So as you can see, the NASH model recapitulates the key aspects of the human NASH disease, and it can be used to predict human responses of anti-NASH therapeutics that mirror the same results of the clinical trials that have already happened. Now, again, we have profiled efficacy of further anti-NASH drugs that are currently in the pipeline. Here you can see Selozertib, Aramcol, Cenicriviroc, SB43142, and Elafibranor, which we use as a positive control. And here is a nice panel of markers that we use to examine inflammation, fibrosis, and steatosis, as we previously mentioned, just a bit more markers in general. And if you see at the heat map, the higher the green is, so the higher the intensity of the green, the bigger the positive effect is of the drug, so to speak. And as you can see, compared to all of them, Cenicriviroc had the highest impact across all of these three different groups of reactions that we want to reduce, so all the other ones had different effects for specific markers. Here on your right, you can also see the confocal images showing a similar image, all having some sort of effect on the different markers, which are here stained, steatosis being Nile red, alpha SMA, and collagen type 1. Here is again the quantification of the imaging, and as you can see, all the drugs had a positive effect in reducing steatosis as well as fibrosis. So collagen being fibrosis and Nile red being the steatosis bit. And again, the concentrations that we used for this experiment were also the clinically relevant ones that were used in clinical trials as well. So these, again, matched the clinical results that are currently happening. Okay, so what did we learn today? We learned that the PhysioMimix NASH assay captures all key aspects of the human disease. We learned that we validated anti-NASH compounds that are currently in phase 2 and 3 clinical trials. We've learned that we've improved the in vitro and in vivo translation, that we're using human translational biomarkers that are relevant to the clinic as well, and that we have predicted that we have created a predictive human model that bridges the gap by supplementing and cross-validating existing data sets to minimize the risk of clinical failures. Thank you so much for your attention. If you have any more questions or any queries in general, feel free to contact me at the email as you see here at the bottom. And I hope you enjoyed the presentation, and good luck with the other presentations as well.